Welcome to today's Explainer. We're about to go on this incredible journey, both physical and historical, into the absolute foundation of biological existence. We look at ourselves in the mirror every morning and just see one unified being, right? But the reality is so much more complex, and honestly, way more fascinating. Today, we're peeling back the layers of life to look at the microscopic building blocks that literally make everything possible.
So, think about it. If we want to define life, we really have to start at the absolute baseline, the non-negotiable requirement for survival. When we look around at the sheer variety of life on Earth, from massive redwood trees to those tiny, microscopic organisms swimming around in a drop of pond water, we have to ask ourselves, what's the common denominator here? What is the absolutely smallest possible thing that can exist independently and still technically be called alive?
To answer that, here is our roadmap for today. We're going to cover 1. The unit of life, 2. Discovering the microscopic world, 3. The universal cell theory, 4. Diversity in shape and size, 5. Plant versus animal cells, and finally, 6. The plasma membrane.
All right, let's start with the big picture. Section 1. The unit of life.
Just like bricks in a building, cells build our physical structure. But they aren't just dead weight sitting there like inanimate bricks. They have all these continuous internal metabolic reactions happening that literally keep us in a living state. If those chemical reactions stop, well, life stops. And the ultimate rule of biology is that anything less than a complete cell simply cannot live on its own. You can't just take a piece of a cell, like, say, a nucleus or a single mitochondrion, and expect it to survive independently. It takes the whole unified cell to pull it off.
And this is where you really see the sheer power and adaptability of the cell. Take unicellular organisms, for example, like a single bacterium or an amoeba. That one lone cell independently performs all essential life functions from eating to dividing. But then look at multicellular organisms, like us humans or higher plants. We have trillions of cells working together, basically dividing the labor to form complex tissues and organs. Yet the fundamental unit, it remains exactly the same. The basic rules of life apply whether you're a tiny population of one or a sprawling, bustling metropolis of trillions.
But how did we even figure all this out? Cells are completely invisible to the naked eye. Let's travel back in time. Section 2. Discovering the microscopic world.
Science is basically one giant collaborative buildup. Back in 1665, Robert Hooke first observed what he called cells in a piece of dead cork. Though, fun fact, he was actually just looking at the leftover dead cell walls. Shortly after that, in 1674, the incredibly curious Anton van Leeuwenhoek used these little self-made microscopes to become the very first human to observe living, moving cells. Fast forward a bit to 1831, and Robert Brown peered deep enough to discover the dense center, the nucleus. But here's the wild part. It wasn't until the 1950s, nearly 300 years after Hooke, that advancements in electron microscopes finally revealed the truly minute details. I'm talking about hidden organelles like ribosomes, the tiny factories responsible for making proteins, which are literally just 15 to 20 nanometers across. No way you're seeing that with an early microscope.
All of these scattered historical observations eventually snowballed into a set of universal laws. Section 3. The universal cell theory.
Here's where it gets really cool. We've got Matthias Schleiden, a German botanist studying plants, and Theodor Schwann, a British zoologist studying animals. These two scientists literally joined forces across their different disciplines. Schwann noticed that animal cells have this thin outer membrane, and he realized plant cells have this extra rigid layer, the cell wall. Together they came out and made this massive unifying declaration. All plants and animals are entirely composed of cells and the products of cells.
Now, their joint theory was a total game changer. It changed science forever, but it had a really glaring hole. They proved what we are made of, sure, but they couldn't explain how new cells were actually formed in the first place. Think about it. If every human starts as a single-celled zygote when a sperm and egg fuses, how do we eventually get trillions of new cells? Where are they spontaneously popping up from?
Enter Rudolf Virchow in 1855. He basically steps up and drops the mic to give the theory its final, perfect shape, with this legendary phrase, omnis cellula e cellula, meaning cells come from pre-existing cells. Virchow proved that life is just one unbroken chain of cellular division. Whether it's through mitosis for growth or meiosis for reproduction, cells absolutely do not magically appear out of thin air. An old cell simply divides to create new cells.
Okay, so now that we know what a cell is, let's take a look at their incredible diversity. Section 4. Diversity in shape and size.
0.1 micrometers. That is incredibly tiny. This number represents PPLO, a type of mycoplasma, which holds the record as the absolute smallest living cell on planet Earth. To put that into perspective for you, it is thousands of times smaller than a single millimeter. But on the total opposite end of the spectrum, an ostrich egg that measures 10 to 15 centimeters, making it the largest isolated single cell in the entire world. It really shows a staggering variance in nature's dimensions, right? From something that requires an incredibly powerful electron microscope just to see, to something you can hold heavily in both of your hands. And this leads us to kind of a golden rule in biology. Form follows function. A cell's shape is entirely dictated by its job. For example, a nerve cell has to transmit signals all the way from your toe to your brain, so it acts like a really long, heavily branched wire. A red blood cell is perfectly molded into a 7 micrometer biconcave disc. Kind of looks like a piece of dough squeezed in the middle, right? And that's so it can efficiently carry oxygen and effortlessly slip through your tiniest blood vessels. Plant mesophyll cells are round and oval to pack in food-producing chloroplasts, while tracheids are these elongated tubes specifically designed to act like plumbing, transporting water up a plant stem.
Despite all this wild diversity, the cells of higher organisms basically fall into two major buckets. Section 5, plant versus animal cells. Now keep in mind, both plants and animals are eukaryotic, meaning they possess a true, distinct nucleus. This is unlike prokaryotic cells, like the bacteria we mentioned earlier, which just have their genetic material floating around without a defined nucleus.
When you look at this direct comparison between eukaryotic plant and animal cells, you really see how each side survives. Plants don't have skeletons, so they need structural rigidity at the cellular level from a thick cell wall. They obviously can't just walk over to the fridge to find food, so they make their own using chloroplasts, and they store their excess water in a massive central vacuole. Animals, on the other hand, we completely lack cell walls, which makes us way more flexible, and our cells rely on a totally different internal toolkit, like centrosomes, to help our cells divide.
Finally, let's zoom all the way down to the micro level to look at the anatomy that makes a cell even possible. Section 6. The plasma membrane.
Every single eukaryotic cell relies on a core triad. First, you've got a boundary to protect it. That's the plasma membrane. Second, there's a semi-fluid matrix called the cytoplasm. This is the main arena for all those metabolic reactions we talked about. It's where organelles live, like mitochondria generating the cell's power, or lysosomes acting as tiny recycling centers to break down waste, all keeping the cell alive. And third, the nucleus. This is the dense brain of the cell, containing your genetic material, organized into chromosomes, basically directing the whole show.
For the longest time, scientists were pretty stumped on how this specific outer boundary actually worked, until they proposed what's known as the fluid mosaic model. It's a game changer. You have to imagine that the boundary of your cells is not some stiff, rigid brick wall. Actually, scratch that. It's more like a highly dynamic, flexible ocean of fluid lipids. And floating freely within this ocean are these solid protein icebergs.
The real chemical genius of this membrane is how it literally builds itself automatically. It's composed of a lipid bilayer, so 2 layers of fat molecules. And these molecules have a bit of a split personality. They have hydrophilic, water-loving polar heads that are super happy to face outward toward the watery environments inside and outside the cell. But their non-polar tails are hydrophobic. They are absolutely terrified of water, so they automatically tuck themselves inward, facing each other. This creates a perfectly sealed, water-repelling core, with those functional proteins embedded all throughout the structure. Because this lipid sea is so fluid and flexible, it gives the cells some incredible superpowers. It acts as a selectively permeable bouncer, strictly controlling what enters and exits. Substances can move passively through diffusion or through osmosis, which is just the specific movement of water based on surrounding concentrations. But if a cell needs to force nutrients in against the natural flow, it uses active transport, which costs energy. This membrane flexibility also lets the cell grow, facilitates vesicular transport by literally swallowing large nutrients, and gracefully allows the cell to pinch inward during division without shattering into a million pieces.
So we've seen today that independent cellular organization, bounded by a dynamic membrane and fueled by internal metabolism, is the absolute defining feature of life. But this leaves us with a fascinating paradox to chew on. Think about viruses. They contain genetic material, sure, but they completely lack a cell of their own. They literally have to hijack ours to do anything.
So, going by the strict rules of cell theory, are viruses truly alive? There's no easy answer, and it's a huge debate that continues in biology today. Thank you so much for joining this explainer, and keep questioning the microscopic world around you.